Transcription is the process of taking a DNA molecule and copying certain sections of it into RNA. And when we're talking about eukaryotic organisms, if we're actually copying DNA and looking at DNA sequence, this whole process is going to take place inside the nucleus. So eukaryotic transcription, the location for it will be inside the nucleus. And we will have a number of different enzymes and proteins involved in this. Actually, with eukaryotic organisms, we do have 3 different RNA polymerases. And each one of those different RNA polymerases is going to be responsible for transcribing a different set of genes. So they will each have their own subset that they go through and they transcribe. We'll just kind of talk about them as a group as we're going through them. And then also with the polymerases, they are going to need some help with this whole process. So these helper molecules are going to be called transcription factors. And we will have a whole collection of these. Some of them will only be involved at the very beginning of the process, and some of them are going to kind of function throughout the whole process until we get the termination. RNA polymerase II is responsible for transcribing the majority of the genes that we have inside a eukaryotic organism. So because of this, we're going to focus primarily on what the promoters for RNA polymerase II look like and just the overall process with RNA polymerase II. Eukaryotic transcription is going to begin with initiation, just like we have with prokaryotic transcription. So with initiation, it really is always about the promoter and really getting the polymerase, and in this, the RNA polymerase II, to the promoter. And so if we take a closer look at what a promoter is going to look like in eukaryotic cells, we do still have a +1 place, and this indicates the transcription kind of start location. And anything behind that would get a negative number, and this direction would be what we would call upstream. And in the opposite direction, we would call that downstream. And one thing that we almost always have in these RNA polymerase II promoters, these regions that polymerase is going to bind to, is we have this region that has a lot of T's and A's, and we refer to this as the Tata box. So this is very characteristic of promoters, and the high quantity of T's and A's here is going to help us to separate the strands a little bit better, because T's and A's only get 2 hydrogen bonds between them. So that will be a region that is going to be easier to actually split apart. And then also, if we were to go even further upstream, more down in this area, we will most likely have a number of regions, and I'm just going to call them boxes at this point. But these are regions that have other sequences that will help to increase the transcription efficiency. So these other regions will be bound by specific transcription factors that are going to help to recruit and get the transcription of this gene to just be a little bit more efficient. So to get this entire process started, the transcription factors, we're going to refer to them as transcription factors 2 because they actually work with the RNA polymerase 2. These are going to help to recruit the RNA polymerase. So these individual transcription factors, they are going to be bound here, several different ones, and I'm going to show them in a few different colors just to emphasize to you that we do have a collection of them that are binding, and their job is going to be to recruit that big RNA polymerase, and it is a big enzyme. So RNA polymerase then is going to come over here as well and join, and once we get all of these together assembled on the DNA, that is what we call the pre-initiation complex. And so the pre-initiation complex, once it is there on the template, then we are ready to actually start making RNA, and that would then lead us into what we would call the elongation process. For the elongation process to really get started, many of those transcription factors that help to get the RNA polymerase to the template and to that pre-initiation complex, they are going to actually release the polymerase so that it can continue on The elongation process in eukaryotic cells is very similar to what we have in prokaryotic cells, in that the RNA polymerase, which is shown here in blue, RNA polymerase is going to be reading the DNA 3' to 5', and as it does that, it will be making RNA 5 to 3. And notice that the RNA is just going to be attached for a short period of time, and then we will see back here that we do have single-stranded RNA that's actually coming out of that transcription bubble. And the RNA polymerase is going to continue along. Now, the overall process of transcription in eukaryotic cells is a little bit more complex because if you'll recall, we do have a lot of different proteins that are associated with organization of the DNA. So remember that we have a lot of nucleosomes, or what we can call histones, that are here with the DNA, and that's what I'm showing right here. And so the nucleosomes, which are made of histones, those really have to be moved out of the way. So we will have to have another collection of proteins whose job is to really clear the template for just a while so that the RNA polymerase can get through. We refer to them as FACT. And what this FACT stands for is facilitates chromatin transcription. Okay, so FACT is responsible for moving these out of the way so that the RNA polymerase can actually get through and it can make the RNA. And then once it gets through, those nucleosomes, they can reassemble with the DNA for organization purposes. With the termination in eukaryotic cells, it is determined by a specific sequence in the DNA. When we talk about termination in eukaryotic cells, this is going to occur with a specific sequence in the DNA. And once the polymerase reaches this particular sequence in the DNA, what we're going to see is that everything dissociates. So what we mean by that is that the RNA polymerase is going to come off of the DNA. It's going to release the new transcript, which is what we would call that piece of RNA that we now have. The DNA is actually going to close right back up again. So this RNA molecule that we now have as a product from all of this, it is single-stranded. It has a 5' end and then a 3' end as well. And this is what we tend to a lot of times call the pre-mRNA. It is not an mRNA molecule yet. We could also refer to it as a transcript. So that would be the product of transcription.